Thanks again, Yuri, for organizing this wonderful session, and I'm glad to be here. So today I'm going to talk about the systems immunology of human immune set points, and I'm glad to follow the guru because I think she introduced some of the concepts I'm going to talk about. So I will try to fly faster than usual. So first, I would just want to acknowledge broadly both my lab. My lab is both a computational and experimental immunology lab, and also the Center for Human Immunology, where we conduct a lot of the human studies and work with colleagues there to develop technologies. So our work, we're quite obsessed with variations at two levels, and also integrating this kind of variations. One, it's the human population level. As you know, human immune responses are highly variable across individual. The other type of variation that we try to integrate in and try to understand is single cells. So for systems human immunology, we ask questions like, why do individuals mount variable human immune responses to perturbations like infection, vaccination, and disease? Can we predict these responses and understand some of the shared mechanisms among them? We also, to address some of those questions, we try to compare sort of human immune system states across health and diverse diseases, both at the baseline, but also in terms of how they respond to different perturbations. And then to do that, we have to build tools, including computational tools, to enable that sort of answering that sort of question. And I won't talk too much about the single cell aspect today, but except to highlight the use of single cell approaches like multimodal single cell approaches to look at the human population. So as I alluded to, and yes, you probably appreciate, when you look at almost any sort of immune response scenario, immunotherapy, response to vaccines and infections, and the extent of or the disease activity associated with autoimmunity, you see a lot of variation across individuals. And even after accounting for certain variables like age and sex and variables like those, you still typically see a huge range of variabilities in how individuals respond. So as you can see here in the middle, for example, here's an example from influenza vaccine responses. You can see the broader, lower responses in the elderly compared to the young, but within each group, in particular, in the younger individuals, you see hundreds of folds of differences in terms of the antibody response. So some of the questions we and others have include, can we predict these responses? Can we uncover potential determinants? Are there shared set point predictors and mechanisms in these different scenarios? So obviously genetics can be an important component, as our age and sex, as the previous talk has discussed nicely, and also the extent of pre-existing immunity, especially classic antigen-specific and B and T cell memory can be important determinants of responsiveness and quality and quantity wise. But genetics alone, it's far from explaining phenotypic variability. So if you look at, for example, influenza again, and you look at adults and look at identical monozygotic twins, the estimated heritability is actually 0, suggesting that genetics play very little role. So if you have two individuals like twins, one can be a high responder while another can be a low responder. And this is likely because of various factors like past exposures, and other types of factors that I'm going to talk about today. So one aspect that we found many years ago now, back in 2014, when we look at influenza vaccine responses in healthy individuals, is that beyond the effects of age, sex, and also pre-existing immunity in influenza, we found that the baseline immune status of an individual can predict the response, and in this case, the antibody response to the flu vaccine. And since then, we have been looking at the surrogate of that signature transcriptionally in both additional influenza vaccine cohorts and also in additional vaccination scenarios, including a live attenuated yellow fever vaccination, and found that to be the case if you measure these signatures, either by the frequencies of certain cells or by their transcriptional surrogates, you can see in a correlation with the antibody response. And so this is a recent paper where we look at this across multiple vaccines, and where we also asked the question whether this kind of age independent sort of set point signature it's applicable in a different scenario. So in that case, we in particular look at disease activity in lupus patients. And in that situation, oops, sorry, where am I now? We basically look at the baseline signatures, pre-vaccination signatures in these patients when they're clinically quiescent. So as if they were sort of like having not a lot of disease activity, and ask whether that correlated with the extent and the quantity of disease activity in a subset of those patients. Whenever they flare up, their immune system looks like they're mounting a vaccine response. And the answer to that question was yes. In the specific subset of lupus patients whose flare has a plasma cell associated signature, if you measure their clinical quiescence state using this baseline signature, we can see such a correlation. But it's very specific to this subtype of lupus patients. If you look at lupus patients with a very distinct type of disease activity, you don't see this kind of signature. So that started to suggest to us that there are these shared set point mechanisms that exist across both healthy individuals, but also in people with disease. And I want to highlight that to figure out what are the cells and their potential states that contributed to that signature, because initially we measured that in bulk, blood RNA-seq. So we use this approach called ssci, which allow us to measure hundreds of surface proteins, the transcriptome, and also the VDJ, TCR, BCR sequences in single cell at the same time. The great thing about this is that we can also use a combination of genetics and hashing barcodes to pool a lot of samples together. So this allows us also to sort for specific, more rare populations like PDCs and other populations that are of interest, to allow us to pool, for example, about 50 different samples, and then conduct this kind of more rare cell type analysis at the single cell level. So because this is a computational immunology session, I also want to point you to some of the computational tools that we had made to look at this kind of data. So here, a project led by Matt and Andrew Martins in the lab, where when we look at the surface proteins in these single cell data, we found specific types of noise that we have to address. So one type of noise we figured out using experiments was the sort of the ambient antibodies being captured by these single cell droplets. And other ones are within each one of these droplets, you also have these background levels of signals, and they came from a variety of sources. And we found that we could approximate that background by looking at a combination of the correlated component in the isotype controls. So check this out, and Matt has done a really nice job of documenting this package and making it very usable across different platforms. So back to the set point signatures I mentioned. So by doing ssci on the high versus the low responders, by doing a comparative analysis, we found that even though the signature is encoded by, we can get it down to as small as like 10 genes, the underlying state that captured by that signature is actually quite complex. It's actually encoded by a network of correlated cell states involving, for example, more activated PDCs. And these are the cells that can actually make more type 1 interferons even at baseline. And then that interferon can lead to a cascade of activation signatures in T and B cells. So it's in these more abundant cells that in the bulk that we can measure such a signature in whole blood. But in fact, we hypothesize that based on this signal at the single cell level, that the origin of the signature is really coming from these PDCs, and they are usually really rare in the blood. And the other interesting aspect of this kind of signature is that if you measure individuals over the course of months, they tend to be temporally stable, meaning that the differences we typically see at a snapshot are being maintained over time. We also have seen certain predictors that seems to be less stable, so they would fluctuate around. But usually these stable parameters, as you would expect, they're the more robust ones that we can detect. Now, since sort of the initial, the flu finding back in 2014, we've also now seen a number of other examples in the literature from both others and also our work with collaborators, for example, from Rafiq's group in terms of HPV in the elderly. There is interesting, actually, they see that inflammation is actually inversely correlated with the response, consistent with what Dagu just mentioned earlier. There's also additional signatures in flu vaccines, as well as more recently, there are some signatures at the baseline uncovered from malaria, RTSS. So now I want to turn this kind of set point baseline signatures onto some other settings. I want to give you a vignette on the cancer immunotherapy front. So this is a collaboration with a couple of NCI colleagues, Zhao Chen and Arang, and together with Matt in my group. So here specifically, we're trying to ask, why do some individuals, after they receive immunotherapy, develop autoimmunity? So here we pick as a pilot people who all of them responded to the immunotherapy. They all had a nice regression in their tumor, but about half of them, in this case 5 versus 4, developed IRAE in myositis, while the rest didn't, basically. And specifically in the analysis, we try to distinguish 3 types of signals. One is what we call treatment-associated signals. So if you see an immune parameter that basically changes after the therapy, so these are signatures that are associated with the treatment itself. We actually kind of wanted to avoid seeing this kind of signatures in a way, because these may be important for mediating the anti-tumor response, for example. So the other one, it's ones where it's similar, but the strength can be different between the IRAE versus no IRAE group. And the most interesting thing for us is this kind of type 3 signatures, where at the baseline before treatment, you can already see differences across these 2 groups, and they are stable even despite the treatment. So they're treatment independent in a way and reflect the intrinsic state of these individuals. And this is actually what we saw at the most prominent signatures of these. We can find quite a number of these type 3 signatures. So here's a more, apologize for the complex plot, where each row corresponds to a gene set signature, and each column corresponds to a cell type. Because we have these site-seq single cell data, we can bin them into these individual cell types and subsets, identify in an unbiased manner, and then ask which one of these show a signature, comparing the IRAE versus no IRAE. So I'm highlighting here one example involving an mTOR-related signature in these CD8 TAMRA cells, and you can see that it's quite clean, at least in these individuals, that you can see this upregulation of this set of mTOR-related genes in the IRAE group versus the no IRAE group. But the underlying biology is, again, likely fairly complicated, because when we look at these different signatures that show a signal, they are quite highly correlated. So you got this mTOR TAMRA signal, and it's correlated with the metabolic as well as various dendritic cell inflammatory states in these individuals. So it looks like this set point, again, is captured by potentially these cells interacting and mediating some of these locking in a way, these states, again, before they even receive the therapy. And this may be potentially responsible for the IRAE response in some of these patients. Now, one of the questions we've been asking over the years is, how are such likely antigen agnostic baseline set point states established, right? How are they being set, and how are they being maintained? So I want to give you an example based on a recent study that Rachel Sparks, William Lau, and Ken Liu led in the lab, where we asked this question of, can mild COVID-19 infections actually shift some of these states, and can that influence a future response? So the reason, one of the reasons why we asked that question is because if you look at COVID, there's hundreds of millions of cases and recoveries, and by and large, the majority of the patients had fairly mild disease, non-hospitalized, basically. So we wanted to understand, for this really large population, what happens afterwards, and are there changes in some of these set point and set point signatures that I've been telling you about, right? So there are some precedents of how infections may impact the immune system. And for example, in humans, measles infections is known to wipe out the B cell repertoire, impacting especially in younger individuals and infants, this ability to respond to a future infection, even though it's not measles. And there's various examples in mouse studies where infection and prior infections can leave an imprint, and that can potentially affect both the status of the immune system and also future responses. And also on the vaccine side, the most well-known case is the BCG, for example, where BCG vaccination epidemiologically, it's known to reduce all-cause mortality in infants. There's also work describing how BCG vaccination may potentially change and train innate immune cells, including monocytes and monocyte myeloid progenitors. There's still a lot of question regarding how long some of these training can last and what their impacts might be for future perturbations. So Rachel really led a clinical team that did the heroic work here during the fall of 2020 at the first wave, after the first wave of COVID, where she enrolled these non-obese, healthy individuals. They basically have gone through clinical recovery. They look like healthy individuals. They had no prior, they had no concurrent disease, and there's no known vaccines given in between when they had COVID and when we vaccinated them using influenza. So these individuals got vaccinated, and we followed them over both the acute response days 1 and 7, and then all the way to 14, 28, and then 70 and 100, and then once every month. We also enroll a matching sex and age match healthy cohort. These were totally COVID-19 negative. They never had COVID, basically, to serve as a control set, basically. So for the rest of the slides, I'm going to mostly focusing on the single cell CySeq data. We analyzed 40 of the individuals over days 0, 1, and 28 to look at how prior COVID, mild COVID, may have changed their. This is right before they got the flu vaccine, and then how it may change their innate response, as well as the adaptive and antibody response. So why influenza, right? So I'm running out, maybe don't have a lot of time. So just to say that we know quite a lot about its dynamics because of our and others' work. And of course, we're supposed to receive the vaccine to help potentially fighting an infection. Although, as we know now, there were almost no influenza during that season, basically. So I'm going to skip some of these issues. And so one of the questions we ask is assessing the differences in the innate and adaptive response. So we basically always compare the prior COVID group versus the healthy, never COVID group in terms of the response. So comparing day 1 to day 0 and day 7 to day 0 and so on, basically. So one thing we noticed early on is that the response is very sex dependent. So we found that when you look at the day 1 response by measuring circulating interferon gamma, we see that the COVID males, surprisingly, had an elevation in the level of interferon gamma on day 1 compared to their healthy controls. Also higher than the COVID recover females and the healthy females. And this is typically you see females responding, especially the innate response is a bit more robust than males. So this was one interesting surprise earlier on. And then when you look at the cells, so this is looking at a single cell level, you do see that all of these, especially in the COVID recover males, many different immune cell lineages in circulation, they mount a strong interferon signature. And this is not surprising given the higher circulating interferon gamma level. So likely these cells are all responding to the elevation in interferon gamma. So you can see this quantification here. And if you look at day 7, so the flu-specific plasmablasts, they're also elevated in the males. So suggesting that the more robust innate response may have led to this more robust adaptive response as well. And we didn't see any difference in the recover females. And finally, if you look at day 28, you do see that across the different strains in the vaccine, maybe except one, but trending in the same way, they were all significantly higher in the COVID recover males only compared to the healthy males, but again, not in the females. So we thought that this is likely because of the more robust innate response. And indeed, we can see that if you look at the day 1 innate response in terms of interferon responsiveness, it correlates nicely with the day 7 flu-specific plasmablast response. So therefore, we focus on asking what is really driving this more robust innate response on day 1, right? What cells are responsible for this elevated interferon gamma response? So by doing an unbiased look using the single cell CySeq data, we found that this marker, GPR56 on CD8 effector memory cells, it's the most differentially expressed surface marker on these COVID recover males. Then when we look at this in a UMAP, so this is marking in a UMAP plotting using the most variable surface markers, the CD56 high cells, we then overlay on top of it a known transcription signature for CD8 cells that are known to sort of so-called for poise for interferon gamma when you stimulate them using eomycin and PMA. And indeed, this overlapped very strongly with that independent transcriptional signature. So we thought that this may be because in the males during COVID, they may actually have a higher elevated inflammatory response. That inflammation may have helped train some of these CD8 effector memory T cells. And so there's this description of these bystander activations of these T cells. So we then overlay this sort of bystander activation T cell signature found in acute COVID with this GPR56 signal. And again, you can see how overlaps. So in the bottom, these are the quantifications of these distribution of cells. So, and indeed, when we look at the baseline of the COVID recovered males, we see that these cells, the 56 positive cells, are at a higher frequency in these COVID recovered males. And when you look at the day one response in these cells, these are the only cells mounting a significantly higher interferon gamma transcriptional response on day one, basically. So with the combination that these males have higher frequencies of these cells, and that these cells make more gamma together, likely contributed to that higher levels of circulating gamma that I mentioned, basically. So I'm not going to, given the interest of time, so there's actually, especially in mouse, there are these reasonably well-studied innate-like memory CD8 T cells. And we think that this is what we're seeing here for the first time, and based on how in COVID, mild COVID may have helped train some of these CD8 cells. And we check a bunch of these surface markers that's seen in mice. We see exactly the same profiles in these 56 versus 56 negative cells, and in how they match quite well with these innate-like memory phenotypes. So our hypothesis is that these are likely trained by IL-15 during acute COVID-19. So with that, I just wanted to sort of summarize and say that, again, the baseline is really, on the one hand, quite malleable. On the other hand, they are being kept shaped by these different infections and vaccination over the lifespan. They can really be established and fine-tuned over long time scales, while in short time scales, they can really impact the response to different perturbations. So this, of course, it also creates a window for us to think about how to modulate these baseline status to improve future responses. And with that, I just want to thank, again, my lab. I already highlighted individuals along the way. And also just want to, some shameless advertisement, I'm in the process of moving my lab to Yale, and also to establish a center. And there will be openings for faculty and staff scientists, and all the way from postdocs to students in computational systems and synthetic immunology. So if you're interested, if anyone you know are interested, please contact me. And it will be launched in the fall of 2022. Thank you.